Everyone, welcome to the Tasmanian Institute of Agriculture's Honours Seminars for 2023. I'd like to start by acknowledging that today we're meeting on Trichloroacetic Tasmania, water, seas and waterways. I acknowledge with deep respect the traditional custodians of the land who have cared for this country for thousands of years. I pay my respects to elders past, present and emerging, and acknowledge those who did not make it to elder status. I recognise a history of truth that acknowledges the impact of colonisation, and I stand for a future that respects and acknowledges Aboriginal perspectives, culture, language and history. Thank you very much for coming today to support our students who are presenting the results of their honours work. I'd particularly like to acknowledge the presence of the Ag Institute, both here and online, who have supported, supported our students for a number of years. And there are also other industry partners, family and friends. Pei, I'd like to hand over to you to introduce the students.
Thank you very much, Shane. Good afternoon, everybody. Thanks for coming. I really appreciate that you find time to come and support our students who have been working very hard over the past months on projects that are solving real-time problems in the agriculture sector in Tasmania and beyond. And today we have about 7 students who will be sharing their journey so far on what they've been doing on these projects and how they've been putting effort to contribute to solving all of these problems. So the students will be having about 15 minutes to make their presentation, and after that we'll have time for questions for about 3 to 5 minutes. And at the same time, this is part of their honor study program. And so there will be an opportunity for you to also assess them. The scoring sheets have been provided via email, but will also be on the screen as we progress with the presentations today. And if you're not able to access the electronic copy, there are hard copies available as well that you can use to score the students, and I'll be able to send them across to you if you need a copy of that. So like I said, this is an opportunity for the students to share the work they've been doing over the past months, and I really believe that these students have been working very hard. I've only come to know them just about 5 months ago, but I can really say that they are very hardworking students, and you're really going to enjoy listening to them as they share what they've been working on over the past few months. So this is recorded as well, and just to let you know, and can be later accessed via YouTube, and the recording is also for marketing purposes as well. So the first presenter for today is Luke. So Luke will be presenting on assessing the feasibility of growing oak trees in Tasmania. So Luke, do you want to come up and...
I didn't bring pens, I can't. That's fine.
So my project was based on assessing the feasibility of growing oak trees in Tasmania to supply the whisky industry with oak barrels. So a bit of background on the Tasmanian whisky industry. It is only quite young, it's only roughly 30 years old, but in that time it has seen a rapid growth, and that growth is projected to increase in the future as well. Given that legally whisky does need to be aged in Quercus barrels to be classed as such, as the demand for whisky does increase, the demand for barrels will as well. The other good thing about whisky barrels from Tasmania is that the industry can reduce their emissions whilst ensuring that production is made in Tasmania as well. So from a review of the literature, the three main species that are used for oak barrels are Quercus alba, Petraea, and Roba. They're all chosen for the different characteristics that they do impart into the whiskey, as well as their ability to be worked into a barrel. Further on the literature as well, the major points were that the global supply of oak current global supply of oak will not meet future demand. So that's a result of poor forest management and a lack of reforestation projects with oak. The biggest challenge in establishing oak stands in Tasmania is likely to be animal browsing, because that can, from development or from the start of establishment, it can completely destroy the whole entire crop or forest, sorry. There was plenty of literature as well on non-timber forest products as well. So in this case, acorns, intercropping with other cash crops, as well as the carbon storage potential that these trees produce, which can all be used to subsidise production costs. So the main questions that we set out to answer were, first off, can oak trees be grown in Tasmania? And if they can be, how big will they grow over time, so that we can assess their carbon storage potential and when we can harvest them sustainably as well. So I went about answering these questions to start with by reviewing the literature to get an idea of how to estimate biomass and growing conditions for oak trees, as well as input from industry. And then I collected data through field work and case studies to build models on biomass. So then we could start predicting how oak trees grow over time in Tasmania. The main conclusions that we came to were that yes, Quercus or oak trees can be grown in Tasmania. The main limitation of this being rainfall and age. By age 100, we can reasonably expect up to 10,000 kilograms of biomass per tree, depending on rainfall, obviously. And then the big thing with this project as well, it does provide the industry with concrete steps on how they can start reducing their emissions too. So how I went about the project, I started off with the National Trust Significant Tree Register. We did find that there was quite a poor age range. They were all based around 75 to 150 years old. Most of them were Quercus roba, so there wasn't very much species diversity as well. And as we can see in the data that we ended up getting from that, most of them were suspiciously exactly 100 centimetres in diameter. So that wasn't quite going to be good enough. So I had to put my boots on, go hug a bunch of trees and find all of these trees around the state as well. So at the same time, I did supplement that data with younger trees from the Tasmanian Arboretum. There was quite a few more species that we didn't get from the Significant Tree Register as well. We did find, however, from all of the Tasmanian data, there wasn't a very wide spread of rainfall. These were the measurements that I took per tree. So the diameter of breast height. The height was measured with the stick method, as you can see in this diagram here, just some basic trigonometry and a stick. Biomass is really difficult to estimate accurately without destructive measures. So given that I couldn't just go lop a bunch of trees down around the state, this equation was the best estimation that we came to. We wanted to compare that data as well to a real world case study. So that's where the US Urban Tree Register came in. This was a data set of over 1,100 trees that we chose, which did show a much better age range and a much better spread of rainfall as well. Again, they were still a little bit limited with our spread of rainfall. We would have liked to have seen more species and rainfall interactions so that we could get an idea of how that impacts biomass accumulation. All of the US trees and the Tasmanian trees, however, were limited to urban plantings. So they weren't necessarily representative of a forest plantation where we would sort of be setting them up in Tasmania, but it did give us an idea of growth rate. And then the climate data was just from the Bureau of Meteorology for Tasmania and the National Weather Service in the US. And then to what we found, from the US data, it was pretty clear that diameter and biomass were very dependent on rainfall. Most of the Tasmanian land where we could potentially be growing these trees ranges from 500 to 1,000 millimetres of rainfall a year. So the data that we found from the US, it did represent Tasmanian conditions quite well. We did find that age as well did have a significant impact on biomass too. As we can see with the R-squared values that we found, 80% of the variance overall was accounted for in this model, with the most significant impact, as I said, being age and rainfall. So from that model, we were then able to start predicting biomass over time. So at 50 years, we could expect roughly 2.5 tonnes extra biomass in a 1,000 millimetre rainfall zone, such as Meander Valley, as opposed to a 500 millimetre rainfall zone where it's a bit drier on the east coast. And then at 100 years as well, we can see that there was a much more significant impact on biomass as a result of species as well. So onto the Tasmanian data, we could see that we found that local government authority and species didn't have a very significant impact on biomass. But again, as was found in the US data, we could see that age did have a significant impact. As we can see here, 80% again of the variance in the model was accounted for with this data. But given that there wasn't a very large spread of points or data points in the Tasmanian data for species and rainfall, we weren't able to use that to model biomass over time. Now, comparing the US and the Tasmanian data, we can see that in the way of diameter, the two do correlate really well. So it is reasonable for us to be able to use the US rainfall data to then assess how rainfall does impact biomass accumulation in oak growth in Tasmania. And then at roughly, or at 50 years, we're expecting roughly 75 centimetres of diameter in the oak trees. And then at 100 years old, roughly 100 centimetres as well. So from the figures that we had there, or from the data that we did find, and the models that we came up with as well, we did have the US, the biomass in the US data, sorry, at the 500 millimetre to 750 millimetre rainfall zones. They did concur really well with 95% of the Tasmanian data as well. So again, from that we can start using, despite species variation, we can reasonably use the US data to assess the effective rainfall on biomass in different areas of Tasmania. So the main limitations of the study that we found were based around error. So again, the biomass methods were an estimate only. Urban trees do behave slightly differently to forest planted trees in the way of competing with each other and soil conditions. The age of most of the Tasmanian trees as well, as I said, were quite old. So we didn't really get a very good feel of how the younger trees behave in Tasmania. And we did ideally would have liked to see more of the more species and the rainfall diversity, especially with Quercus alba, where we had quite a few older, higher rainfall Quercus albas, but not very many low rainfall. So we weren't able to accurately assess the effective rainfall on Quercus alba. For the industry to use a project like this, it gives them an idea of whether or not it's worthwhile setting up a trial. And if they do decide to set up a trial, whereabouts in the state we can set up that trial, and what limitations need to be assessed and addressed beforehand. It also gives them a concrete plan of emissions reduction as well. So at the end of the day, planting trees is pulling carbon out of the atmosphere. So if all else fails, they've at least got that under their belt. That takes us to where future research should be directed, using that wider rainfall and species diversity so that we can get an idea of how the two do interact and how that impacts biomass accumulation. Gauging how other soil, geographical and climate factors impact biomass, particularly drought and waterlogging. Validating those biomass equations, so ideally having more local Tasmanian oak biomass equations, as well as using oak trees that are grown in plantation settings as well. The other thing is exploring how hybrids can be used to achieve higher biomass in shorter periods of time, and whether or not this is going to impact the trees in the point where we can or can't use them for cooperage. And then thanks to Anne, who's here today, and then everybody else who's listed above as well.
Thank you very much, Lee.
You were first.
Was I? I had a couple of questions. With, pruning, so you've got these urban trees which you talked about a little bit. There are no pruned, sort of thin oak growing areas or orchards in the world that could have been compared to?
Is there what's that?
Is there any orchards of oak trees where they're grown them for bats and furniture and things?
There were.
That you could get a better, because you talked about the problem about the urban situation, and I agree with you with pavements and different hydrology, where you could have got some understanding of how they would differ.
Yeah, we didn't, because ideally, we would have been able to compare the difference between the two.
Yeah.
We did find though that the ones that were, because you have truffles and things like that as well that you can use. I did find that they were using like the conditions that they were growing the oak trees in. They weren't growing them straight up like you want with in cooperage.
Oh,
There was dwarfing trees, and then there was, like you said, pruning, things like that, where they weren't necessarily growing in the conditions that we want them to. So yeah, it was sort of splitting the difference. Ideally, we want some forest setting crops or trees, but the data set was big enough that it sort of evened itself out, if that makes sense. Well, that was what we came up with.
We've got a few questions, so maybe I'll skip my second one.
Yeah, so you mentioned this a little bit just a second ago. Is there any chance you can, the truffle industry and the cooperage industry could work together or be part of the same industry? Yeah, so that could happen.
So the tricky thing, I was going to talk about truffles initially, but I left it out because they're, as lots of people will be aware, they're quite tricky.
Yep.
So yeah, that is one thing that they can use them for. It would obviously just be ensuring that you're prioritising the tree rather than truffle production.
Yep.
I guess, and not sacrificing growth from the oak trees.
Matt?
Hey, I should probably preface this by saying I'm actually a forester. This is what I do for a living. I have a couple questions. Firstly, did you look at altitude impacts at all?
No.
Okay, because altitude can have a really big impact on timber quality.
Yeah.
So that's going to limit a lot where you'd be able to grow this sort of stuff. Because like straight off the bat with the rainfall stuff, I'm thinking, oh, we could go, up in the northwest, but that's high altitude, but it's high rainfall. So, if they can't tolerate the altitude or the timber quality drops, then that's not going to be suitable as well and limit some of the more traditional forest growing areas. Second question is, what's the oak like with soil? So what are their soil requirements? Because we have quite acidic soils. I don't know what the soil types are like in Europe where they grow normally.
Yeah.
How would that, do you know at all how they would tolerate that sort of soil environment?
I did find that alba, that's probably going to be our target sort of species, given that it is the most resistant to those sort of soil conditions. Ideally, you don't want to stick them in a waterlogged acidic soil because they will really start to struggle. But generally, if you stick them in a well-drained soil that's not too waterlogged or anything like that, then yeah, generally they're pretty hardy to those sorts of things. I'm not 100% sure about the other two species though, off the top of my head.
Yeah.
Addressing your first point as well, we were more focused on already cleared agricultural land, so generally the flatter sort of neutral, I guess, altitudes.
Yeah.
So yeah, that is one thing that we would ideally like to go into, but sort of beyond the scope of the study.
Yeah.
Unfortunately.
I suppose if you could link it up with like a truffle sort of business model, then that would work quite well. But I know from personal experience, that ag land is at a premium and it's getting very hard to put any trees on that sort of land, and a lot of it's being converted back into agricultural systems. So especially for a very long rotation thing like you're looking at with sort of an oak stand, like it's going to be a struggle getting that land to go into trees unless you have some sort of other business model that you could use. Because yeah, like 75, 100 years is a long time to wait before you get anything merchantable off a stand.
We could just take two more from Nathan and Dougal because they raised their hands first. And maybe take one more online, then we'll move on to the next. Sorry.
Yeah. My question's pretty short. Do you think it's worth industry people doing a trial? You said that was one thing they could get out of this. Do you think it's worthwhile?
I do, because it does have flow-on effects for Tasmanian industry. So obviously it gives Tasmanian industry barrels, but it does also promote sort of barley growing in that land as well. And yeah, sort of the flow-on effects of what it can do for the industry too.
Last question.
So I was thinking about quality, because obviously the whiskey's pretty high quality and pretty expensive stuff. And slower growing wood tends to be more dense and presumably have effects on the oxygenation in the barrel, and also maybe some of the flavour and aroma impacts it has on the product. So I suppose follow on a bit from Nathan's question, if you were to recommend a trial, normally we can't just put 100 trials out in the field, we might be lucky to get one or two. Would you be going for a slower growing, lower rainfall type zone, or would you be going for a faster growing, less dense, timbered product? I'm just interested in your thoughts from the literature review, I suppose. Yeah. It's not related to your research.
Yeah, I mean, like you said, that's obviously going to have a significant impact on the product, the end product. With a trial and touching on the cost of land and things like that as well, those areas that are lower rainfall that aren't going to be priced as high for agriculture, they're probably going to be your better options. And then you can get an idea of, you can sort of extrapolate from that data as well and get an idea of where to go next. So you're not trying to obviously get it right 100% perfect the first time. It would probably be better to go for that lower rainfall sort of area. And you do, as you said, get denser wood as well, which is probably better for whiskey.
Just take one last question from you.
Yeah, most of the other people asked the questions that I was going to ask. Other than in your literature review, did you get a feel for the economics of this?
Not really. That was one thing that we wanted to look into further, but again, we were sort of limited to one major area. So we did touch on that a little bit, but yeah, it wasn't a massively in-depth sort of area.
I'm just wondering what the cost benefit is, whilst there's this sequestration and all those things, versus you're talking very, very long time.
Yeah.
It's extraordinarily slow, but the whiskey people assure us that it's they want to do it, so they take the long-term view.
That's a long PhD to do this one.
Just one last question online, and then we'll move to the next speaker.
The questions, you showed a correlation with rainfall. What was the minimal rainfall required?
For oak tree growth? Again, it does depend on the species. I don't have the exact figure for what was required for those on average for the species.
Thank you very much, Lee.
Thank you.
So the next student up is Jacinta. Jacinta will be talking on the effect of environment on hard seededness and seed softening in serradella.
So a lot of people aren't familiar with what this review looks like. So I'm just going to pass this around and you can have a little touch and have a little look. So thank you for being here today and welcome to my presentation. My presentation will be on the effect of environment on hard seededness and seed softening in serradella. So serradellas are an annual pasture legume. They are a nutritious legume with high palatability for all livestock, and serradellas can be used for grazing, hay, or silage. The seeds develop in the canopy of the plant, and they have a range of key attributes. They are highly suitable to sandy and sandy loam acidic soils with high levels of extractable aluminium. They have a deep rooting establishment compared to subterranean clover, which can lead to an extended growing season. And they also have a lower phosphorus requirement compared to subterranean clover. So these serradella species have a potential to fill a niche agroecological gap in southern Australian pasture systems. So serradella seeds have physical dormancy. This means that the seeds have a water impermeable seed coat, and this dormancy mechanism is known as hard-seededness. So hard-seededness is an important survival mechanism. It can prevent germination and seedling death in out-of-season rainfall and assist in pasture persistence. The breakdown of hard-seededness is known as seed softening, and this is the physiological change where the seeds become permeable to water. There are substantial variations in the initial hard seed percentage and the rate of seed softening among species and among cultivars. And another interesting physiological feature in serradella species is their pod. So each serradella seed is enclosed in a protective hard pod, as illustrated in the slide here. Okay, so moving on to some background of the bigger project. My honours supervisor, Rowan Smith, is currently running a serradella project on 12 different serradella cultivars. So my honours project is investigating 4 serradella cultivars. These are Yellow Tas Avila, UTAS 1050, UTAS 1046, and I've also looked at 2 subterranean clover cultivars, Goulburn and Lyra, as my control. And then also bringing your focus to the table here, we see that there's variation in seed softening within the same species. And there's also unknown information for these 2 pink serradella cultivars. It should also be highlighted that some of the serradella cultivars are developed specifically for Tasmanian pasture systems. For example, the UTAS 1046 cultivar has been selected for the later flowering, and this is of high interest to Tasmania as we generally have a longer growing season. So moving on to my research questions. My first research question was, is the seed softening rate of cultivars influenced by the environment? Is the initial level of hard-seededness influenced by the environment in which the seed is grown? And does seed softening vary between cultivars? Okay, so I've got some baseline information that I covered. First, we assessed the seed and pod characteristics under the stereo microscope and moved on to looking at capturing high quality images of seeds under the environmental scanning electron microscope. And lastly, we obtained the seed and pod weight. So looking quickly at our baseline results, we found that the Avila and Yellow Tas, their pod was similar in their rectangular shape, and they also had fine hair-like structures over the surface of the pod. In contrast, the UTAS cultivars, the pod was much more rounded and it had the tapered ends. And looking at the surface of the pod, it appears much smoother and it does not have any visible hair-like structures. And here's just a side-by-side comparison of the 4 serradella cultivars that I focused on, and this is just a good snapshot of each serradella seed. Okay, so for the serradella seed pod weight, we found there was a significant cultivar effect on the seed pod weight. So for the yellow serradellas, they were much heavier than our pink serradella cultivars in terms of the ones that I focused on. So major experiment one, slide one for major experiment. We found, so first we harvested the seed and then we placed 100 seeds on a Petri dish with 3.5 mils of water. The seeds were placed in the germination cabinet at 15 degrees Celsius for 14 days. And at day 7 and at day 14, we counted and removed the dead and the germinated seeds. At day 14, we found the initial hard seed percentage. So we found this by firmly pressing down on each seed and confirming if they were either soft or hard. So continuing on with the major experiment materials and methods, we placed the seeds in nylon flywire pouches with 100 seeds in each pouch. And we had 2 main sampling points at 3 and 6 months. So this meant that the pouches were left on the soil surface exposed to the environment until the testing occurred. So then at 3 and 6 months, the hard seed percentage was obtained. Okay, so I have 4 environmental combinations. My first environmental combination, the seed was grown in Tasmania and softened in Canberra. For the second environmental combination, the seed was grown in Canberra, softened in Tasmania. For the third environmental combination, we had the seed grown and softened in Launceston. And then for the fourth, the seed was grown and softened in Canberra. So for the environmental conditions of Launceston and Canberra, there were some noticeable differences. So for temperature, Canberra was generally warmer throughout the late spring and early summer months compared to Launceston. And then for the monthly rainfall, Launceston received the majority of rainfall over the winter months and early spring, while Canberra was almost the opposite. We also found the 2023 Launceston soil surface temperature highly fluctuated between January and April compared to the Canberra site. Apologies for that, it's slightly squished the slide. So moving on to our major experiment results. So here are the hard seed softening results for 2023. So on the x-axis, we have our sampling points at 0, 3, and 5 months. And then on the y-axis, we have our hard seed percentage. And then a quick reminder, we've got Launceston grown, Canberra softened, Canberra grown, Launceston softened, Launceston grown and softened, and Canberra grown and softened. And I'm going to bring your focus to 4 of the key points in my figures. So firstly, apologies, the slides are slightly squished, I'm not sure why, but so for Avila and Yellow Tas, we found that there was a very high initial hard seed percentage. However, Avila illustrated very little seed softening over the 5 months, while Yellow Tas had a significant softening in the first 3 months of environmental exposure, regardless of the environmental combinations, except for the Canberra grown Launceston softened seed. We're not actually 100% sure why it didn't follow the trend. So moving on to the third point, the UTAS 1050 cultivar had a moderate initial hard seed percentage at about 60%, and it remained hard for the 5 months of the experiment. And for my fourth and final point, we have UTAS 1046, which had very little softening over the 5 months. You'll also see that for the UTAS 1050 line, it slightly went up, and we think that that is from the slight variation in the seed samples, because we're not testing the same seed at 0, 3 and 5 months. It's different seed, but it's from the same seed lot at the start of the year. And then for the 2 subterranean clover cultivars, they followed a known seed softening pattern, and this indicates that the experiment ran correctly. So after my 2023 findings, we were interested to know if this initial hard seed percentage and the softening pattern actually, if it was similar or different between years. And this was possible because my two of my supervisors, Rowan Smith and Rebecca Haling, generously provided the 2022 data for my project. We found that there was a significant effect between year, location, and cultivar. As we can see, yellow ties clearly has the highest level of seed softening out of the cultivars I tested. Yellow ties also demonstrated a quicker softening rate in 2023, and we suspected that this was due to differences in climate between years, such as the higher soil surface fluctuations during 2023, and this may have led to a sped up breakdown of the hard seededness in the seed. And then for the UTAS 1046 and 1050, there was no major changes between years. So key findings, Avila and yellow ties have known softening patterns. So Avila, high initial hard percent, it remains hard. For yellow ties, high initial hard percentage, and it softens quickly. For UTAS 1046, it follows a similar pattern to Avila. And then the UTAS 1050 is unique in terms of the cultivars that I assessed, as it has a moderate initial hard seed percent and has little to no softening over the 5 months that my experiment ran for. And there were significant differences between years. However, the cultivars continued to follow a similar pattern. So Avila never acted like yellow ties, and vice versa. So implications, there were many important, there are many opportunities to manipulate management and optimize different serradella cultivars for long-term pasture persistence. For example, we have UTAS 1046, and what we could do is sow a combination of scarified seed and pod. So the scarified seed would germinate in the first year, and then at the end of the first year, it would drop the mature pods on the soil surface. And then the seeds that were sown remain hard in the first year, but potentially germinate in the second year. So therefore, with appropriate manipulation and of serradella cultivars, such as this one here, it can lead to pasture persistence and lower susceptibility to out of season rainfall. So here's a couple of future research. We could research the potential effect of seed coat and thickness and pod thickness. We can assess the long-term seed softening pattern of the 2 UTAS cultivars. This could be over like a 2-year time frame. And we could combine knowledge of seed softening patterns and flowering time for selection of the appropriate cultivars in the southern pasture systems. So a big thank you to everyone on this slide. Thank you for your assistance, guidance, and help throughout my project. Thank you for listening. And do you have any questions?
Nathan?
So perhaps I wasn't paying attention, but I was just, there's a good chance that it's my fault. But why does it matter if the seeds go soft? Why is this something you're actually interested in measuring?
It's very important to know if the seed softens or not, because from a farmer's perspective, if you sow all these seeds and nothing comes up, that farmer's not going to sow, or it's likely that that farmer's not going to look at serradellas again. So knowing that softening pattern of the seed is very important.
Right. And the softening pattern is related to the germination, is that what you're saying?
Yes.
Yeah, sorry, I just didn't get that from the start.
Mike?
I was wondering why you chose to use Launceston and Canberra, and you didn't choose to do it all in a growth chamber with, you know, with a lot more control of light and temperature and so on.
So the choice of Canberra and Lonnie is from the bigger project, so that's where they're doing the testing. And in terms of doing it either in a controlled environment compared to the actual environment, I think, well, doing it in the actual environment might represent an easier, it might represent
Realistic
a
conditions.
realistic
Yeah.
outcome. Thank you.
What was the difference in the environmental conditions between 22 and 23?
So I had a look at them, and for the 2022, the rainfall distribution was slightly different. So can't quite remember specifically, but for one of the sites, there was like an additional 60 mils over the first 6 months of the year. In terms of the soil surface temperature, we actually haven't quite looked at that yet, but we will look at that because it would be interesting to know if the 2022 soil surface temperature, so if there was more fluctuations or less between years of the soil surface temperature.
Because that was the other bit I was going to ask is
Yeah.
in the literature, is there much information as to whether the trigger is moisture related or temperature related or an interaction of the two?
So it's a combination, and it's actually the fluctuation is really important. So in the literature, you can see that, well, I've only covered about 5 to 6 months, but in the literature, when you see between years, there's actually this pattern where you have high levels of seed softening and then it drops down, and then into the next year, it goes back up again. And that frame of the high seed softening is generally where we have the higher fluctuations in temperature.
Last question, please.
I'm going to take a punt here because I may have missed the detail. This is a leguminous species.
Yes.
Okay, that matches with the photo I took of it. So one of the major benefits of this plant then is that it grows on these acidic aluminium toxic soils. Is that right? So that we can, because my understanding is a lot of leguminous species require a higher pH soil or like higher levels of pH and calcium and so forth. Is that the main reason for the using of species?
Yeah, because there are areas where we would like to grow legume species such as subterranean clover, but because they don't survive very well in these acidic conditions, we need to find something else.
Okay.
And it is a better option, or it is an option to have these this type of legume compared to other forms of changing the environment such as mining.
Thank you. I understand now. I was studying the photo, I think, when you might have explained that. Thanks.
Thank you.
Thank you, Jacinta. I know that most people here have the physical copy to score the students, but for those joining online, you can follow the link to score the students or scan the QR code. So our next speaker is Cara. Cara will be talking to us on understanding the role of biological control methods in Tasmanian apple orchards.
thank you everyone for coming along today. So as Ate said, my project looked at understanding the role of biological control methods in Tasmania. So pest management, as I'm sure you're all aware, is an ongoing challenge in agricultural production systems. One of the key pests of apples is codling moth. Codling moth, or Cydia pomonella, is native to Central Asia, as is the apple tree. It's been present in Tasmania and in many present in Tasmania since the 1850s due to international apple trade, and is found in most apple production systems worldwide. When left unchecked, codling moth can cause damage levels of up to 100% on apple trees. And one of the reasons why it's such an important management issue is because of the low export thresholds associated with using apples and so sorry for the changes in the slide. I think that seems to be a consistent issue. But some of the challenges with managing codling moth in orchards is related to conventional control and the associated effects of that. So we've got chemical resistance, loss of insect biodiversity, secondary pest outbreaks, which is where pests which were previously controlled by natural enemies, which I'll explain later, their populations can then become more abundant, and other issues including labour and fuel costs associated with controlling cotton. So I'd like to introduce integrated pest management to you. Integrated pest management aims to promote biodiversity in the farm, and by encouraging the role of natural enemies and minimising the effects of chemical controls. Integrated pest management is the intersection of cultural, biological and chemical control. My project focused a lot on biological control, so I'm going to break that down further for you. The main aspects of this are classical, augmentative and conservation biological control. If we look at the first two, they relate to the use of natural enemies in the orchards. Now, natural enemies are organisms that will control pest species in their natural range and can include pathogens, predators and parasitoids. You might be familiar with the first two, maybe not so much parasitoids. They are species that require a host to complete their life cycle, and in doing so kill the host. So parasitoids can be very effective natural enemies because they can selectively target specific pest species. Classical biocontrol is where natural enemies are released in the long term and populations establish, whereas augmentative biocontrol involves the supplement, is where these populations are supplemented. However, you cannot introduce natural enemies without supporting them through conservation biological control. This can be done through the planting of forage ground cover, and it provides nectar and pollen, which can support insect populations, increase their lifespan and their activity. So my project looked at Mastrus ridens, which is a parasitoid wasp of codling moth. Mastrus, which I'll be referring to it because it doesn't have a common name, is native to Central Asia, just like apple trees and codling moth. So it was collected from Kazakhstan in the 1990s for the purpose of biological control in apple production systems worldwide. Parasitism rates of up to 70% have been reported for Mastrus, and it's been released into countries including California, Chile, New Zealand, and Australia, to name a few. It's been released in Australia since 2013. However, mixed population establishment success has been recorded, which is especially the case for Australia and New Zealand. and something I will be talking about more. Just to give you an idea of how codling moth and Mastrus interact with one another, as I said, parasitoids can be quite specific, as is Mastrus, so it will only target codling moth in the larval stage when they're cocooning. When codling moth cocoon, they supposedly produce an aggregation pheromone. This lets other codling moth know where they're cocooning so that they can commune together. The reasons for this can be speculated. Yeah. So the aggregation pheromone is released, and from our understanding, Mastrus ridens can detect and respond to that. And it's understood that it's attracted to 3-day-old cocoons of codling moth to the greatest extent. To break the aggregation pheromone down further, I should also point out that this aggregation pheromone is different to the codling moth sex pheromone, which is produced by codling moth adults. So this is only produced in the larval stage. Codling moth larvae will respond to the aggregation pheromone produced by other larvae when these compounds are present. However, Mastrus ridens requires a more complex composition of compounds to be released. We can also just point out that these results were derived from a study that collected pheromones from 300 codling moth larvae that were aggregating together. And then they took that pheromone and did further chemical analysis. So just keep that in mind, that's what they based it off. So why has Mastrus ridens population failed to establish in places such as Australia and Tasmania? Well, one of the key reasons is inbreeding. The population was collected almost 30 years ago now for the first time, and all releases up until end of last year in Australia derived from the same population of 400 Mastrus females that have undergone a number of genetic bottlenecks moving between laboratories worldwide before coming into Australia. And that affects the fitness and sex ratio of Mastrus. We can also look at the laboratory rearing conditions and how this has affected the Mastrus, particularly if they respond to pheromones from the larvae, and Mastrus in the lab have been fed larvae which subsequently have been fed an artificial diet, then this may not relate to larvae in the field that have been consuming wild apple. The time of year is also very important. Mastrus will only fly when temperatures exceed 15 degrees. So in order to ensure establishment, we need to make sure that the time of year is appropriate for these conditions. Looking for a solution, there's been a new genetically diverse population collected from Kazakhstan once again in recent years, and we can ask whether this could help provide greater establishment levels in Tasmania. So my research questions were: 1, is there a chemical difference between wild and laboratory read codling moth aggregation pheromones? How will the new Mastrus ridens population behave in Tasmanian apple orchards? Does Mastrus ridens have a preference between wild and laboratory reared codling moth larvae? However, spoilers, we weren't able to test this. This was meant to be a bioassay with Mastrus, but we observed a very low emergence rate of the Mastrus culture. So we weren't able to do that. Instead, the question became, why did the new genetically diverse Mastrus ridens population fail to emerge? My 4th research question was, how can biological control be supported in apple orchards through mixed forage ground cover? However, due to time constraints of today's presentation, I won't be covering that. So I collected wild codling moth larvae from suburban apple trees around Hobart because these are less likely to have chemical and cultural control, and larvae will be more abundant. We placed them into plastic, ventilated plastic containers with corrugated cardboard sheets because the apples would contain codling moth larvae. When they would emerge from the apples, they would seek cocooning sites, so we provided them in the cardboard. We replaced the sheets every 10 days for 40 days. And you can see there's a codling moth cocoon in the cardboard. And we also recorded how many larvae we observed to be emerging between these periods, and found that we could have started collecting the codling moth much earlier, because in late March from onwards from there, the number that we emerged per period essentially halved. So this means that the number of fresh or 3-day-old cocoons will be more abundant in March. So to my first research question, we sourced lab-bred codling moth larvae from Ag Victoria through the PIPS program, and I got those larvae and the ones that I had collected in the cardboard sheets. I forced them out of their cocoons and got them to respin. This is to standardize the age of the codling moth cocoons, and then subject them to gas chromatography, mass spectrometry, and expose spiny fibers overnight to see the. So what we found from the mass spectrometry looked at the composition and the presence and concentration of compounds in samples. And we found that none of the known aggregation pheromones that I mentioned earlier were present in any of these samples. There were 2 common compounds in each of the samples. And we also noticed that there was a qualitative difference between compounds produced by lab and wild larvae. As you can see here, 2 compounds that were present in all lab samples and in none of the wild codling moth larvae. However, these were not aggregation pheromones. Statistical analysis was not possible due to a small sample size and the long time that it took to conduct this analysis in the lab. So how will the new mattress riders population behave in county apple orchards? We conducted a field release to test this. It was on the 24th of May. We released parasitized codling moth rollers in a central location and set up sentinel traps, which are rolls of larvae in further locations to test whether the mastrous wasps were emerging from parasitized cocoons and moving through the orchard. We retrieved traps on the 24th of August and incubated them for 2 weeks at 25 degrees, and then dissected larvae to see if any indication of parasitism was present in the sentinel traps. However, upon doing this, I found no evidence of successful parasitism by mastrus. And we can ask why this is. There's a bunch of different reasons. One of them is, as I mentioned, mastrus will not fly above 15 degrees. So if we look at the number of days where the maximum temperature exceeded 15 degrees in the last 10 years in Grove, which is the nearest weather station, we can see that for the period that we released the mastrus, very few days, in some months 5 days, were suitable for flight or development. So this does not ensure that establishment has its best chance in the orchard. As I mentioned earlier, we got a lot of codling moth moving out of apples into cocoons in March from the Hobart results. And you can see here, March in Grove has almost every day of the month suitable for mastrous flight. So we can think about how this would be a more supportive date to release mastrous. To my last section for today, we want to know why the population failed to emerge when reared in the laboratory. So this was for the bioassay test, which didn't go ahead. We reared them at 20 degrees for 48 days. It was meant to be much less than this, but we extended the period because we only got 5 mastrous out of 200 codling moth to actually emerge as adults. So to see why that was, I dissected the codling moth larvae, and I did find indications of parasitism within the codling moth. Examples of that are below. We have a mastrous adult cocooning mastrous within their own smaller cocoons and heavily consumed codling moth larvae. And then on the right, a codling moth head where a codling moth has emerged from without parasitism and a codling moth pupa. So what we found was that of the 197 codling moth larvae we reared in the lab, only 10% of them showed signs of mastrous inside of them. And then of the mastrous that were present there, only 11% of them emerged as adults. So I had 89% of mastrous still within the cocoons, but dead, which is particularly concerning, especially because if we released the mastrous into the field and only 10% of those were parasitized, and assuming these results can be translated, 11% of those emerged, that's not giving us enough chance to establish mastrous in the field. So in summary, chemical compounds detected are not supported by published literature. A clear difference in compounds produced by wild and codling moth larvae was observed. Mastrous released in May will not emerge throughout winter months in Tasmania, and low parasitism and emergence rates were observed in the mastrous colony. In the future, we should consider releasing mastrous in March or earlier to ensure establishment, re-examine rearing practices in terms of inbreeding and genetic diversity, and investigate composition and concentration of aggregation pheromones further. Thank you, big thank you to everyone who supported me in my research, to John and Steve, my supervisors, and especially Fruit Growers Tasmania, who supported me through the Fruit Growers Tasmania Honours Scholarship. Yeah, thank you.
Any question? Can we take 3 questions maybe because of time? Michael?
Me?
Yes.
Yeah. History is littered with invasive species coming in, and they either take over or they like a damp squib and they fail. So I guess my first question is, how did you know that the codling moth wouldn't just take over and it would be another gorse or digitalis or blackberry, or, you know, I mean, that would be horrendous.
Yeah, sure. So one of the main benefits of mastrous is that it's a parasitoid. So that means it can only effectively complete its life cycle when it targets codling moth. It doesn't reproduce very effectively in many other hosts because it's evolved together with codling moth. And there's been specificity testing done every time mastrous has been released in a new location, and that was done in Australia in 2013. And they found that it couldn't effectively reproduce on other species.
What role does, you talked about temperature, but does wind and sunlight play? Because you obviously got to pick up these pheromones as well as being able to fly towards them. Is there any knowledge about that? Because we've got different conditions to where this thing came from.
Yeah, I don't have any information specifically on that, but wind is definitely a major influence on insect flight and behaviour. So yeah, that would have to be taken into consideration. And we did have a lot of windy days this year.
Yeah, that's what I was thinking
So
about.
yeah, it's something to consider.
Yeah.
I'll take last question from Ian and then one online.
Apologies if you said it and I missed it, but is there any potential link between the gases that you found and the parasite not being able to locate it? I mean, is that a particularly interesting finding, or?
So we weren't able to test if it was able to locate it.
But you sort of did because it's only 10% of the.
Yeah.
Sorry.
Wait for the
Is it
answer.
a potential explanation for why the parasite isn't going to work? It was because the gas given off here is different.
Yeah, potentially. Yeah.
How will climate change influence the survival of the mastras?
Yeah, that's a really good question. Something that I can't entirely answer to a full extent. Mastras will often show its best fitness at 25 degrees and 35 degrees lifespan, and other aspects as including how many eggs it lays will be reduced. So it is something to take into consideration, especially more variable weather will definitely affect it. But yeah, not something I can
Thanks, Karen. So our next presenter is Emmaline. Emmaline will be talking on phosphate solubilizing bacteria in Tasmanian soil.
Good afternoon, everyone. My name is Emmaline, and I did my project on phosphate solubilising bacteria in Tasmanian soil. Phosphorus is a fundamental macronutrient with extreme reactivity, and it likes to bond to other compounds to become biologically unavailable. Phosphorus is crucial in the formation of ATP, proteins, and DNA. And it's been found to have deficiencies have been found to cause low germination and plant growth and disease in animals and humans. In the soil, phosphorus exists in both organic and inorganic forms. Inorganic forms exist as minerals, adsorbed and bonded phosphorus. This exists in equilibrium, and the equilibrium has a tendency to go towards the bonded and unavailable forms, whilst organic phosphorus comes from biotic sources and contributes up to 80% of total soil P. This is usually more biologically available, but it goes into the equilibrium and it likes to bond tightly with other compounds and then not be bioavailable. Tasmania has the highest proportion of Australian ferrosols, which like to covalently bond due to their low pH and their high levels of iron to phosphorus and lock it up and make it non-bioavailable. They're widely used in agriculture, and their strong bonding means that lots of fertiliser is added, which exacerbates the bonding by adding more phosphorus. You're adding more to their equilibrium and pushing it towards the non-biologically available forms. It's really hard to get soils to push back towards biologically available forms, and ferrosol is notorious for locking up phosphorus. Chromosols are less reactive with phosphorus, and they're from predominantly southeastern Tasmania, and they have a different mineral structure and a higher pH and higher organic matter. PSB, or phosphate solubilising bacteria, come from a wide range of bacterial taxa and produce strong organic acids which help free up this phosphorus in the soil. They make up to 20 to 40% of the microbiota and help encourage the bioavailability of phosphorus to plants. They cause localised pH reduction, produce organic acids, enzymes, and various isomers of phosphatase, which can help free up this locked up phosphorus. My research questions were varied because what I was trying to do was work out if we had PSB in Tasmania and then see how they could be used within Tasmanian soils. So my first job was to isolate the PSB from Tasmanian soils and to see if these isolates, when used as an inoculant, had an impact on growth by freeing up phosphorus. Were combinations of these isolates more efficient in helping germination and unlocking the phosphorus within the soil? And could microdialysis be used as a novel tool to measure the impact these PSB inoculants had on freeing up the phosphorus within the soil? My experiment was split into two parts, one to isolate and determine the most efficient phosphate solubilising bacteria, and the second part was how to utilise these phosphate solubilising bacteria. So for the isolation, I collected soil samples from 5 different sites that were under different management techniques. I had a market garden, bushland, a paddock, to name a few. This was to give me a diverse range of bacterial species, hopefully, and these were then plated onto Pikovskaya's agar, or PVK, which contains phosphate. The phosphate solubilising bacteria, by producing organic acids and enzymes, free up the phosphorus within the agar and create zones of clearing. So these isolates that had success at freeing up this phosphorus were then characterised. The morphology, the enzyme activity, acid production, and the solubilising potential, because obviously different strains may have different potentials in different things, were assessed. These were then identified by sequencing when I found efficient strains. This here is a halo production within the agar, which means it has solubilised the phosphorus that's in the agar and produced a zone of clearing. By the addition of bromocresol purple as an indicator to my agar, I was able to determine organic acid production. Here I have a localised production of organic acid around the colony with the yellow and the purple. Then there's a paler yellow, and a vibrant yellow was considered strong organic acid. If the agar remained purple, no organic acid was considered to be produced. Isolates had different degrees of solubilisation, so I was trying to cull from a lot of isolates down to efficient isolates. Here I did an enzyme assay to check what phosphate release through the production of phosphatase. Some isolates had a linear increase with phosphate release over time. Other isolates, however, produced phosphate release and then stopped. This might be because of an inhibition reaction with the substrate from the reaction. So further isomers of phosphatase create different things. They all significantly produced higher phosphatase in an alkaline environment at pH 9. For the combined potential to get the best isolate to use as an inoculant, the light blue one here shows the actual halo production of each individual isolate within a 3-group mix. The dark blue line, however, shows what the actual result was when I plated them at the same cell concentration in a mix on the agar. This indicates that some of them had much higher than expected development of phosphate solubilisation, and these were considered phosphate solubilising efficient isolates, which I then named consortia 1 to 8 and used them as my inoculants for my seeds. This comes to the second part of my experiment. So to test my efficient isolates in a real life situation, what I did was got the isolate, fine-tuned it down to get the best isolate for the job, and then mixed them together and I knew what they should expect it to do. I then sterilised some allium cepa or spring onion seeds, which is a main crop in Tasmania, and planted them into ferrosol. This was a random block design. There were 4 replicates of each trial, and they were in a glass house for 29 days. I observed germination, plant growth, and the soil-soluble P of the ferrosol that they were planted in. The time of the trial, and because of time constraints, meant that plant growth may not have been optimum because they can take 14 days plus to germinate, and 29 days isn't really an accurate representation, so a longer trial would be needed. What I found most interesting about this experiment, as you can see here, I have some that didn't germinate and some that did. The control of no inoculant didn't seem to do very much at all, but then the varying consortia seemed to have a significant effect on how the plant grew. Consortia 1 had the highest germination, but not that great growth. Consortia 5 had a lower germination rate, but the most significant difference between the groups for growth, which was biomass and plant length. Consortia 2, 3 and 4 had same germination amount, but Consortia 2 had significantly higher growth at day 29. Consortia 6 germinated, but didn't form any biomass, they just kind of stopped. And 7, 8 in the control had the same germination amount and very low plant growth. I then, after the plants were harvested, I then used microdialysis to check what the phosphorus level was in the soil. This was very interesting because Consortia 5, which had the highest growth, had the lowest soil phosphorus concentration. So whether the plants utilised it, still to be found out. The control with no inoculant had the highest levels of phosphorus within the soil. And the conclusions from my experiment were that yes, PSP can be isolated from Tasmanian soils, and they are very similar to what is found in the literature and have similar organic acids and enzymes produced. The isolates did work best in consortia to grow spring onions, and there was significant differences between inoculated, non-inoculated, and the combination of inoculants used. And microdialysis did give me feedback on how these inoculates freed up the phosphorus from the soil and impacted the soil concentration. These results were very promising, however, so much research needs to go on to determine what organic acid is produced, what isomer of phosphatase, how they work, which ones are the most efficient, which ones work most efficiently together. It also means that we can determine whether the bacteria work best as a consortia on the seed with further research or in the soil. Do they work on the seed to help the plant free it up, or do they work best in the soil to make it bioavailable to the plant? And what effects does fertiliser impact have on this? Do they need to work with fertiliser? Can they be used alone as a biofertiliser, or do they work best with the fertiliser? Does this mean timing and application of fertiliser and fertiliser type is relevant? And finally, I'd like to thank Shane, Bianca and John and the tech team and all of you here for listening. Thank you.
All right, 1, 2, 3 hands up. Jan?
Oh, yeah. Did you sterilise the soils at all that you used?
No, I had a ferrosol sample that I think Richard had got for Holly's thesis a few years back. I didn't I got the chromosol, my source was from chromosol, which was active, and then I planted it into the ferrosol, but I sterilised the seeds and then inoculated the seed. So I have done further trials, and PSB are capable to be isolated from the ferrosol I use, but it seems to be morphologically what I put in is what I'm getting out. But I didn't actually run any analysis on what was actually in the soil. It was more to isolate some, pick the ones with the best phosphate solubilising potential, and then use them to see if we could unlock some of that ferrosol phosphorus.
Yes.
Okay. Did you look at if the phosphorus that was getting solubilized, if that got into the plant at all?
Further study would involve actually checking what the phosphorus concentration was within the plant. Yeah. I basically use biomass and plant length as an indicator of growth, but most studies in the literature have gone into breaking down how the plant's taken it up, what the yield of the plant is, where the phosphorus is in the plant, so whether it's in the leaf, the root, the stem, and how the bacteria have changed the levels. There has been studies have found that you can reduce your fertiliser use by about 50% in some cases with some crops in some soils using these bacteria. But a lot more research needs to be done because there's been nothing in Tasmania so far. So we weren't even sure, although we hoped, because most soil contains phosphate solubilising bacteria, what they were, or if they were there, or how well they work, because Tasmania's got unique soils and it's also got fairly unique climate. And it's got the distance from everywhere else. So it does give you that kind of little isolated place.
Yeah, with Did you understand or look at what type of bacteria that these?
I did. I ended up with species were Pseudomonas, Klebsiella, Sphingobacter, and Antioa, which are consistent with phosphate solubilising bacteria within the thing. I also came across, on my plates, I had 2 fungal species, which were very, very good at phosphate solubilisation. But because I was looking at the bacteria side of things, I didn't use them further through my experiment. But mixing, like Aspergillus, for example, is a fungal species with really good phosphate solubilising potential. Mixing that with some of these bacteria may give a fantastic result because they work together to sort of synergistically help each other. So one might produce an acid that the other one favours as a source, and then others might produce an enzyme. And they seem to give each other like a cooperative kind of approach to getting that out of the soil.
Because that's what I was wondering, whether when you referred to the isolates, they were pure isolates or whether they were.
They were pure isolates, which I then I started off, I had about 80. And then I culled them down through by checking their phosphate solubilising potential, if they produced acids, if they produced phytohormones like indole acetic acid, because phytohormones can help the plant grow. So it might not necessarily be the phosphate that's helping the plant grow, it might be the other compounds. So each bacteria seems to produce a unique combination of phytohormones, acids, enzymes, and different strains, like isomers of enzymes. So you've got phosphatase, which breaks down phosphorus, but there's dozens of different varieties. So which one is better? Which bacteria produces it more efficiently at certain soil conditions? So there's a lot of almost like environmental within the bacteria, within the soil, that impacts how the bacteria work and what they turn on and off. Because like I found with the phosphate release, once they reached a certain level of phosphate production, phosphatase production, they switched off and stopped doing anything. So there's a lot to learn about isomers and like the organic acids. There's different strengths of organic acids to which one works better. Yeah, so there's a hell of a lot, Lamish, to learn about.
Is it time for one more? No.
We can quickly take your Pacific shot.
Based on farmers' strong interest in biological health measures.
Yep.
With your work, I saw you used a colorimetric method.
Yep.
Do you see any potential of developing a simple farmer or field-based test for measuring the capacity of different soils to?
They have actually, in the literature, there are lots of tests, especially in countries like India, where finance is an issue, where they can do, farmers can do on-field tests without having to send their soil off somewhere to get it analysed, which is a cost constraint. So they can do like a field test kit, almost like a swimming pool kind of, you put your thing in there.
Yes.
But yeah, and then there's all the other all the other soil tests to check what phosphorus is. So microdialysis is very efficient in a lab because it's a fairly compact machine. Like you can put it out in the field, but it's very delicate, and it's sort of, I use very small tubes, which meant that the probe and the perfusate was going into basically a large proportion of my sample. Whereas if you put it out in the field, I'd imagine that you'd get fluctuation and variability with your results. So you'd need to use that with other field sort of tests to make sure you were getting the accurate result.
Thank you. We might talk to you more about that.
Definitely.
Thank you very much, Emmaline.
Okay, thanks for coming. My name's Tom Thiessen. I was investigating mechanical pollination in onion seed crops this year as my onions project. Firstly, why look at mechanical pollination in onion seeds? Well, it's a very high value field crop. It has both national and global significance. And in Australia, our production's dominated by hybrid configurations, where that's the use of a genetically male sterile with a pollen donor line. So it's reliant on mostly insect pollination to transfer pollen. With that reliance, it's mainly through the use of managed bees and also feral honeybees that are around, and that causes some inherent risk in itself. We know that yields can be susceptible, particularly in onion seed crops, as a factor of seasonal variation, particularly in weather. We also know that honeybee foraging behaviour isn't conducive to the hybrid configurations within those crops. And there's also just other inherent risks, particularly in the light of how varroa mite is impacting us at the moment. And so it's important to sort of look at those. alternative pollination methods. Again, why are onions so good? They're a great starting point for a lot of the other vegetable seed crops. This is particularly because of their uniform canopy, so it makes it easier for our harvesting and delivery protocols. Also, they're quite a high pollen abundant crop, produce a lot and have a bigger window as well for pollination. And also that hybrid production system where we know that there's no self-pollination available in those flowers. So if we can isolate those heads, we know through mechanical pollination that we're applying the pollen and isolating it from other pollination factors. And also it's got a binucleate pollen, which is more robust and more robust to our sort of cruder experimental protocol. So what are the components? Well, we have to harvest the pollen first, as you can imagine. So for that, I used a modified cyclone backpack vacuum, which was used to target single heads that then traveled pollen through to a cyclone to separate plant debris from it, and then ended up in a small reservoir where we could get a pollen sample. Then had to process the pollen, which was using a fine sieve where I can just hand brush it through. That's removing more of those sort of plant debris parts, particularly the anthers, which are the things that release pollen. Then for storage, because I was using pollen soon after my harvesting, I was storing it over 48 hours maximum on silica beads for desiccating it. There's longer term storage, particularly in freezing temperatures. And then before putting in a delivery system, it undergoes a rehydration to get it back to a viability level that we want. And then of course, the aim is to deliver it well. And that's not an onion crop as it usually, but the idea is to be able to largely do it in a large field scale. So, so for delivery, that was where most of my research was conducted for the year. I did that in 2 different trial sites. So firstly, for delivering pollen, it's important to have a carrier. The factor of that is that we want to dilute the pollen source because you're not necessarily having a large amount, you want to make it more scarce and be able to exploit it across a larger range. So for that, we use 2 different carrier types or 2 different categories, that being of liquid, which is suspending it in a solution that contains an osmoticum, and then also in a dry carrier, which is just straight into a dry powder. They both have their positives and negatives. For the liquid carrier, it's easier to meter the pollen in the applicator, largely due to the already available spray technology in agriculture that's more conducive to liquids. However, there are some impacts on pollen quality as well that we should be aware of, mainly being that when it's suspended in a solution, we have an osmotic potential in that solution, which can cause the pollen grain to either swell and burst or conversely desiccate and dry out. So getting that range is right. And there's also potential interference with a liquid carrier to interfere that pollen and stigma interaction necessary for fertilisation. As for a dry carrier, it's less easy to meter in an applicator, largely because of the lack of dry powder applicators available. But it doesn't run into those problems that of impacting pollen viability that you do with the liquid, as you don't have to maintain an osmotic potential. So my first trials found me, unfortunately, in Myrtleford, a very nice area. This crop was a hybrid crop, so it had that male sterile line with a pollen donor line, where we could isolate single heads to know that the pollen that we were applying on was going to be the only pollen that were making it to those flowers. We explored several different types of carriers. We used a 1:100 weight to volume ratio, and they were all 6 AM applications. That's from previous studies have shown that that's the best time for application. For the seed set, we looked at doing several different applications over time, so we could target a longer receptivity window of those flowers, with the pollen being harvested and used generally within 24 to 48 hours after harvesting. Main measurements we wanted to assess were pollen viability in suspension through in vitro process, how that carrier type affected the pollination rate, and then how it went on to affect fertilisation and seed set. So through the in vitro screening process, one wanted to look at how those different carrier types affected pollen germination on BBK agar plates. The assessment included counting, well, we applied the pollen to the plate, then it underwent a staining of aniline blue, where it could then be fluoresced and looked at under a UV microscope. And we wanted to count the number of viable pollen grains as well as the number of non-viable to know what's germinating and what's not. The viable determined by the growth of pollen tubes. We looked at a range of different carrier types. The main ones were polyethylene glycol, long chain polymer, sucrose, mannitol, another plant sugar, and calcium nitrate were the main ones, where we did 2 different concentrations to reach that target osmotic potential. Interestingly, had some data as well on pollen assist, which is a commercially available pollen carrier, but it didn't actually result in any germination at all, likely from it not reaching that target osmotic potential. And it's available for another crop type, so different pollen grain size, different requirement. But in terms of the success of it, we found that polyethylene glycol on several different occasions performed as well as the no carrier, and in some occasions actually performed better as well. Whereas the remaining 3 sort of were non-statistically significant from each other, but still were able to achieve somewhat pollen viability. So how did it result in the field? We narrowed down some carriers when we were actually testing in the field. We found that we were able to achieve a large pollination rate across different flowers. However, the rate of pollen they were actually depositing on the flower was fairly low, between about 4 and 8. As you can see, this is a sample from one of those stigmas where you often get some at the surface, but some pollen grains end up going down the style and sort of missing the point where they need to be for interaction. Compare that to a hand pollination, which was applied using paintbrush straight to the stigma surface. We're able to achieve a much higher rate. So, how did it affect seed set? It didn't. We weren't able to improve any yield, improve any yield through the use of liquid carriers in this trial. Probably a factor of that low deposition rate matched with low pollen viability in the suspension. Interestingly, we actually resulted in seed set in an unpollinated control, which confounds it in itself. So we had to question what was going on there, and potentially some robustness issues with the hybrid varieties, as well as maybe the isolation bags affecting the microclimate around the head. So we still move on. Next, we went on to dry carriers. So for that was down here. It was based in out at Cambridge, Richmond, and that was on an open pollinated variety. So that is capable of self-pollination, transferring the pollen to its own stigma surfaces. And to isolate the flowers to make sure that we weren't getting any cross from the same head, we had to remove flowers around the targets and also remove the anthers on the flowers that were the targets. The application process was different via paintbrush tip, but we also wanted to assess the same sort of things we assessed in liquid carriers. The in vitro screening process produced some good results. We had a variety of different types of carriers: xanthan gum, sterilized brewer's yeast, and lycopodium spores, which are deactivated spores of the club moss. They all perform similarly well. Interestingly, the xanthan was not statistically different from no carrier, which is important, whereas then it started to taper off with some of the others. Again, in the field, we saw a range, we were able to achieve quite a wide range of pollinated stigmas, around 80%, 80 to 90%, but we're not getting that target. We're not getting the high pollination rate we want to be able to affect seed set. That range between about 1 to about 5. Regardless, when we actually used it to test seed set, it was a bit of a win, especially compared to the liquid, where we would know that unpollinated control didn't result in any seed set. Still, none of the carriers were necessarily significant from each other, but they were comparable to a raw pollen control. And as you can see on the right, they're actually quite low seed set results. But given our pollen viability, so that sort of about 20 to 20% range, with actually quite a low pollen deposition rate, our maximum potential seed set was still only low. So that gap that you see between the bars and the maximum potential is quite realistic given our protocol. And that's accounting for the physical barriers that a viable pollen grain has from reaching the stigma surface, making it all the way down the style, and then fertilising ovules. So we're quite happy with that. I think that's a success, but we need to apply more pollen grains to the stigma surfaces. That's our major limiting factor. As you compare it to an open pollinated crop, open pollinated flower, so that was not isolated, exposed to the availability of insects, selfing, wind, they were able to achieve or exhibited about 24 grains per stigma surface. So quite shy of that, obviously. And given the pollen viability around 20%, we want to be able to get up to that 24 grains or even higher. And that's represented here in a tissue clearing test that we did where we examine actual pollen tube growth down the style. So in an insect open pollinated flower, you can see many pollen tubes growing down with the potential of fertilising the 6 ovules available in an onion flower. Compare that to one of the dry carriers where we just about saw 1 or less throughout it. So the main thing is getting more pollen grains to the stigma surface. So what are the next steps? As I've said, pollen viability could vary fairly often. So understanding the environmental factors and even plant factors that go into affecting that is important, not only important for mechanical pollination, but improving yields in general in onion seed crops. And also, how can we improve that pollen adhesion rate? Well, there's several factors that are there to look at. The role of electrostatic charge, particularly. We know that in natural pollination, bees gain a positive charge through active flight. They pass that charge on to pollen grains, giving the negative charge of flowered surfaces. There's a quick adhesion rate that occurs in natural pollination. And then from there, given what carrier types we sort of narrow down, what are the application technologies that we can use to use that in an open field? I'd like to conclude by thanking Claude Innovation, a large sponsor of this project, as well as SBS, who provided the field trial sites, and of course my supervisors, Al, Ryan, and Cam, and the rest of the Seed Purity team. Thank you.
Two questions. Let's start with John.
That was really good, thank you. Do you think you could get more pollen grains onto the stigma by spending longer time applying them? Like, could you spray them for longer? Would that work?
Yeah. So like that seed set application, we actually did do about 3 applications across 6 or 7 days, just to get a longer period of receptivity.
Yep.
We also did explore concentration of pollen in that. It didn't actually affect it a whole lot, or relative to the amount of pollen you actually have to put in to improve, which is not worth it necessarily given the labour costs and stuff.
Oh, yeah.
Yeah.
Given that slide with the electrostatic forces, that's pretty interesting. Could you not apply that to your process, like electrostatically charge the material?
Yeah, we did actually have a go at that. I think the limitation was the applicator. And also understand the charge needed for certain particle size is very dependent on that size of the particle. So exploring potentially a range of different charges on that onion pollen would be necessary. But we did try with one with just an on and off switch, which didn't improve it at all. Yeah, but yeah, definitely is the next steps, I think, for increasing adhesion.
I was wondering also about the 6 AM. You talked about 6 AM.
Yeah.
Was it with the chemical application, right?
Yeah, with the pollen application.
So why 6 AM? Bees aren't active at 6 AM, are they?
No, they're not, but just from previous results, we found to have better, I think it resulted in better seed set.
Obviously, it's going to be very still then and cool.
Yeah. Yeah. I don't know what it affects of the biology of the plant, but the interactions you are only having, I guess, are between the applicator and that plant surface. So yeah,
could be
just had to choose a time, really.
That's what I'm wondering.
Humidity, maybe.
Yeah.
So the pollen's hydrated And you need, when it's in a liquid carrier, you don't want to dry it back out again. So you actually need it to stay in the carrier on the plant and germinate beforehand. So you're applying early when it's not drying out so quickly.
Okay, thank you.
Ian, did you have a chance to review academic literature in general, and what, if you did, what evidence is that similar techniques actually work at all? Like,
Yeah, on other crops and stuff as well. Well, it's known to be used commercially for a couple of crop types. Kiwi fruit from New Zealand.
well, I did it 30 years ago.
Oh, yeah.
Total pain in the butt. But
Yeah, well, there's
is
that.
it actually promising, or is it just
I think
fancy
so.
tech?
I think it, especially like there's potential for it in maybe where pollination is more limited. That's where you want to be filling the gaps. I know it's done in some tree nut crop types. It's done in date palms and a bit of lettuce as well. And it holds potential for breeding programs as well.
All right. Thank you very much, Tom.
Cheers.
And finally, we have Zoe coming up to talk on the application of Trichloroacetic to promote flowering in multi-resistant Zoe.
Okay, so my name's Zoe, and I'm going to be talking about the induction of flowering in carrot using trichloroacetic acid, TCA. So carrot seed production makes up a large component of the Australian vegetable seed industry, with around 1,400 hectares grown across the country, worth an estimated $35 million per annum. These varieties are mainly proprietary varieties that are grown for international vegetable companies, and they can be either hybrid or open pollinated systems. So naturally, the carrot is a biennial plant, meaning it needs 2 growing seasons separated by a chilling period. But for seed production, we grow it in 1 season, usually sowing in around late summer, early autumn, and harvesting at the same time the following year. But to get there, they need to pass through several stages of development. The first one is the juvenile stage, and this occurs between sowing and when the plant becomes sensitive to chilling. It ends around 8 to 12 initiated leaves, and it's just focused on vegetative growth and establishment during that stage. Once it becomes post-juvenile, it will be sensitive to chilling and it will enter its fertilisation stage. There's 2 main factors that plants require for fertilisation. One is the temperature of chilling and the other is the duration of chilling. So it varies between varieties, but most require a temperature between 0 and 14 degrees for 1 to over 10 weeks. Once they've got that fertilisation requirement and spring brings warm and long days, stem elongation or bolting is promoted, and so is flower bud initiation. So bolting and flowering are significant issues for vegetable producers, as it can greatly decrease the quality of the root. So as a result of this, we've selectively bred against bolting and flowering, so we've bred for bolting resistance. However, this has then caused extra challenges in the seed industry, as carrots now have these really high requirements for flowering. And it also limits the places that we can grow carrots, especially in our milder winters we have here in Australia. An example of this is in South Australia, there's quite a large scale dryland production of Carota, a tropical seed. And to align with the natural rainfall there, the seed is grown in June. And this creates a very cost-effective system, but you're also limiting that period possible for fertilisation. So without any management interventions, it's not a reliable or viable growth system. So what are the management options? Dealing with insufficient fertilisation is not many options to choose from. So the first is artificially chilling roots or stecklings. This involves taking the roots into cool stores for their chilling period and then transplanting them back into the paddock. However, this is not logistically or economically feasible. There's high costs associated with getting that much space in a cool store and all the transport costs as well. You could also extend the growing season, so make sow earlier in the season, but you're moving into the warmer months where establishment isn't going to be favourable. And it's also locking up land for longer, so it's not viable for farmers or seed producers. Then we come to probably the most attractive option, which is using chemical to artificially manipulate flower induction. Now, this has been done before with a chemical called Dalapon or 2,2-DPA. It's a grass herbicide that induces flowering, and it, after commercial use guidelines were developed by Seed Purity, it was used in carrot crops all over Australia, but it was unfortunately discontinued by manufacturers in 2018. So ever since, there's been a major effort within the industry to find a replacement, and screen trials at Seed Purity identified that TCA was a very promising alternative. It's the most structurally similar compound we have to Dalapon, as you can see from these diagrams, and applications in field trials have increased bolting and flowering rates significantly, with no negative effects on the seed yield or quality observed. However, the guidelines for its effective use are not known. So we know that TCA increases flowering, but so it has to be affecting one of these three processes, whether it's the length of the juvenility, the cardinal temperature for vernalisation, or the duration of the chilling for vernalisation. But we don't know, and if we can find this out, it'll inform us how we can most effectively use TCA, when to apply it, and the range of locations that we could potentially grow carrot seed in conjunction with TCA. So my project aimed to use controlled environmental conditions to identify whether TCA reduces the length of juvenility, the upper temperature limit for vernalisation, or the length of vernalisation. And we also compared the efficacy of Dalapon to TCA over all the experiments. This is just a basic overview of what all of my experiments followed. So we used two varieties of carrot. First one is Corota, a tropical type, which has moderate flowering resistance. And then the Nance, which is a European type, has a lot higher flowering resistance. And Nance is an industry standard we use here in Tasmania. So after planting around 5,000 seeds into pots, they were transferred to the glass house where they underwent their establishment and initial growth. The chemical treatment was sprayed before they went into chilling, and then after their allocated chilling time, they came out back into the glass house where they entered the grow out period. They were scored for flower initiation 30 days into the grow out period, and you can see that photo there is when they were scored positive for flower initiation. You can see that first flower bud forming, and they were scored until 90 days on a weekly basis. So these are the three experiments that we designed. The first one is the length of juvenility. So in this experiment, we grew the carrots out to different ages before we treated them with the chemical. And this was measured using thermal time, which is essentially looking at the accumulation of heat on a daily basis. So the lengths we chose were 800, 1,000, 1,200 and 1,400. They then went into chilling temperatures of 4 degrees for 12 weeks. In the second experiment, the length of vernalisation, we applied the chemical at the same time, which was 1,400 growing days. They went into a chilling temperature of 8 at varying durations of 4, 6, 8 and 10 weeks. Then the final experiment looked at the cardinal temperature or upper temperature limit, and once again grown out to the same stage, sprayed with chemical and put in for 12 weeks at 4 different temperatures, 10, 12, 14 and 16. Now the ranges of treatments that we chose were based on previous literature, and we wanted to include an optimal treatment, but we also wanted to include a marginal treatment to see if we could see that TCA reducing or replacing the requirement when it was marginal. So these are the results. This is the first experiment, the juvenility experiment. So in the Corota, I'll tell you what's going on. On the x-axis, you have the thermal time treatments, and on the y-axis is the total percentage of flowering carrots. So at 800 growing degree days on the far graph, can see that there's quite a low bolting percentage, flowering percentage, but it then goes up with higher thermal times. So this suggests that the end of juvenility is probably likely to be somewhere under 800, but the optimal time for juvenility would be up around 1,000, where it's getting those high bolting rates. Then in the gnats, you see there's nothing going on at all at 800, and then some bolting at 1,000 and some bolting with the other treatments. So we can conclude from this that the end of juvenility is around 1,000 growing degree days. Optimal, probably around there too. Now you would expect that once it gets post-juvenile, that line would increase with increasing thermal time and you'd get up around 100% bolting and flowering, but we didn't. And we think this is likely to marginal chilling factor that I'll discuss later. So these are those same graphs, but with chemicals added onto them. The orange is the 2,2-DPA or Dalapon and the grey is the TCA. So in Corota, you can actually see the chemicals decrease, they had lower bolting rates than the control. This was because when we applied the chemical at this age, the plants were too young to withhold that chemical damage. So a lot of them were severely affected by that. And then in the higher thermal treatments, there's no difference. So this is pretty significant evidence that we're not shifting the juvenility. In the gnats, we saw a bit more action. So nothing happened at 800 growing degree days, which we've established was marginal. But once we get up to 1,000 growing degree days, there seems to be a response to the chemical. But because we're not seeing anything happen when the juvenility is marginal at 800, and we've already concluded that this lack of bolting is probably due to marginal chilling, there's not significant evidence here that it is shifting the lower rate for lower requirement for juvenility. Okay, this is the length of fertilisation. So you can see in Corota, 4 and 6 weeks were marginal, but once you get up to 8 and 10, that was sufficient for high flowering percentages. In the gnats, none of them were sufficient, and unfortunately didn't see any flowering in the control. Now with the chemicals added, in the Corota, you can see that the chemicals have a higher percentage of flowering over all of the different lengths of analysation. The difference is most pronounced at those shorter durations, especially 6 weeks. So this is suggesting that when the length of analysation is marginal, the chemical accelerates the response to whatever duration there is available there and accelerates flowering percentage. And once you get up to 10 weeks, there's no effect of the chemical because natural induction is enough. And in gnats, you see a similar trend, except there's nothing happening at 4 weeks because that's way too marginal. And then the TCA and Dalapon are increasing. And the upward trend of this graph probably suggests that the natural duration for induction is way out here at past 16 weeks or something like that, which is a lot larger than we thought it was going to be. It's the final experiment at upper temperature limit. So just looking at the right-hand side of the graph of the Corota, you can see that there's nothing happening at 16, a little bit happening at 14. Therefore, the natural upper limit's somewhere between there where it can still vernalise. And we didn't see anything in gnats once again, probably due to a lack of the duration that we gave it. Okay, so in the Corota graph, once again, looking at the same position, you can see that there was no increase in flowering at the 16 degrees, suggesting it's not altering that upper temperature limit. There's a little bit happening at 14, but the TCA is actually lower, so nothing really happening there. Interestingly, in gnats, the DPA did induce bolting when nothing else did. And so that is some evidence that the DPA has shifted the upper limit slightly. But yeah, TCA is still 0 there. So what can we conclude from all that? The most compelling evidence is that it's reduced the duration requirement for vernalisation. We can see that over both varieties. Could have maybe had something to do with the upper temperature threshold in the gnats, but maybe probably not. And there's no evidence it affects juvenility. Therefore, we can conclude when the best time for application will be, and that's going to be post-juvenile, but before chilling begins. And you probably saw over all the graphs that unfortunately the Dalapon was still much more effective than the TCA, but there's still potential for TCA to be commercially useful, especially in those corroder types where we saw a really high response. And we probably were a little limited in the study with the Nance variety that we did use because it had a lot higher chilling requirement than we thought it was going to have. So from these results, we have been able to calculate some optimal application dates. So this little table here is for Nance, which is covered up, but using data from Cressy in Tasmania and based on the juvenile period of 1,000 growing degree days that we established in the first experiment. So yeah, by accumulating the day degrees from sowing date, we were able to come up with those dates. And then just a tiny bit more on the corroder production where TCA is probably going to be most useful. So in South Australia, there's at least 300 hectares that's completely marginal, and TCA definitely appears to have potential to help with that. And once again, we've done the same sort of table. So further work from here, this project really highlights how TCA works and underpins the usage pattern for TCA in inducing flowering. And in combination with previous trials and more large commercial trials, we could definitely look at getting it approved for use in carrots, as well as predictive models to predict when and if TCA will be useful. And those are just my acknowledgements. I'd like to thank Al, Gracie and Ryan, my Utah supervisors, as well as Cam from Seed Purity, my other supervisor, and support from South Pacific Seeds, Bejo and Seed Purity on this project.
Thank you very much, Zoe. One question for Zoe too. Anyone? Okay.
I might have missed it, but did you apply the TCA at the same previous commercial rate?
Yes.
DPA? And
They're different to the DPA.
Yeah.
So it was at 5 kilos, whereas the DPA is at 3 kilos per hectare.
why is that?
That was from what we'd observed in the previous field trials, that it was a bit heavier and had some leaf damage when applied.
Because we're wondering as to whether you're able to get it closer to the DPA with rate, but obviously not. And is it with an adjuvant?
Pardon?
Is it with an adjuvant?
No, it's just applied as in its sodium salt form.
Yeah. Yep.
Do you think it's worthwhile designing an experiment where you investigate the interactions between the three different things? Sort of where you're varying the rates and the so you've got three different sort of explanations of why it might work, and then you investigate each sort of separately.
Yeah.
It could be. Do you think there's interesting interactions where it's going to work with a particular setup? And
I think if there's interactions between the temperature of vernalisation and the length of vernalisation, it's still probably going to work in the same way. If we apply it at that post-juvenile stage, it will have the time to react to those. So I don't think practically, but it could be an interesting, interesting study.
Right, that's all we have for Zoe today. Nothing online. Right. Thank you very much. Last but not the least for today is and he'll be speaking on early development of industrial paper waste, peat moss replacement in containerized seedling.
Yeah, so this is I basically wanted to sort of look at maybe solving like a sustainability need for a business, and this project was funded by Timberlands Pacific, and this is an industrial paper waste that's produced locally at Norski's Gog Hoya in the Derwent Valley. So why would we want to replace peat? It's not really a renewable resource. It stores probably twice the carbon in the world that trees do. It's also a significant component in the hydrological cycle in the areas that it's in. It has a massive ability to hold heat water, and you can see it being strip mined in there. This can lead to an ecosystem collapse, not just in this area, but also forested uplands that might draw on this water. And in a timberland specific context, they planted 1.1 million radiata seedlings last year, and that's approximately 83,000 litres of peat. This little grass here, this was supplied to me last year by Hilton Henning from Pinion when I was looking at alternative growing substrates to peat, and this was grown exclusively in this waste material from Norski, and to my eye, it looks like a pretty happy plant. It's got good root contact, it looks quite sticky. So this industrial waste, I'm calling it secondary pulp sludge material, and it's a cellulose material, so it's a combination of a linear and branched polymer with the lignin removed, so it holds a heap of water, 80% weight on weight, and it also has nutrients in it that are left from the processing of the pulp by Norski. There are a few things that have to be remediated with it, so it carries quite a few phytotoxic compounds, hexavalent chromium being one of them, and it also, because of the high water content, it has a really strong ammoniacal smell. It's quite, yes, it's not great. It smells pretty bad. So it requires a secondary composting to address that phytotoxicity. This is just a life cycle assessment of this here. If you just quickly look at the picture, this is a Norski. This is the boiler that sort of produces this material. But starting back with these white boxes here, this is the feedstock and thermomechanical processing of this raw material. That's removing the lignin to just leave behind the cellulose, which gives your fibre your paper its strength. Moving through into the green boxes, they're starting to add different chemicals. Sodium dichromate as a bleach is one of them. And there's also some different products in there that will help brighten the paper depending on what's being made. This flows from various stages of this process into a waste stream, where they're basically using these clarifiers to separate solids from the wastewater from processing. Probably the key sort of step is that the bioreactor here, it's heated to 65 degrees, oxygenated from the bottom, and it contains urea and phosphoric acid to attract microbes in to digest the material and reduce its carbon content, so that it can be disposed of more easily. And the little red box down the bottom is our SPSM being produced. This is the current method of disposal that's being used just above the Plenty River. You can see these sort of long lines here. These are long composting windrows. Norski produce tons of this material every day, and this is the current method of disposal. And after composting, it is primarily given to dairy farmers with a safety data sheet to help improve organic matter on their farms. It's currently undergoing an EPA investigation or prosecution because of salmon pond deaths that occurred further downstream in 2020. You can see the river snaking just at the bottom there. And there's also complaints from landowners downstream that are concerned about soil contamination because they've been drawing from the Plenty River to irrigate. So I've got 4 research questions. Can SPSM be treated quickly and safely to produce plant biomass in comparison to the old method, which takes 9 months or to a year to sort of break down? Can chemical, physical and biological techniques reduce the leachate in the growing system? And how does the mycorrhizal inoculation affect the partitioning of chromium from that sodium dichromate in plant parts? And finally, a summative question is SPSM a viable replacement for peat moss in production nurseries? So this material coming from Norski, they were generous enough to supply me with quarterly chemical analysis for a couple of years prior to this project. And it's highly variable depending on what they're making and the inputs they put in to produce the final product. So these green boxes, your pH and carbon nitrogen ratio are quite stable. And for growing, this carbon nitrogen ratio is actually pretty close to ideal, which makes the additional processing complicated. You don't want to push too far and you don't want to necessarily leave it where it is. And these red boxes are wildly variable depending on what they've put in. Well, one of the concerns with radiata, it might be good to have high nutrients in a material that you're growing. The nurseries that produce for timberlands don't actually fertilize at all, the radiata, they just grow it in 100% Baltic peat. Because too much nutrient in there is going to let the plant develop too quickly. It's going to become nice and sweet. And when they put it in the field, it's going to get eaten. This is my sort of method to contain what might be an environmental issue coming from the old method. So I've put them into 150 litre rotary compost tumblers. They're registering every 20 minutes. They're sitting on a load balancers, just made from Arduino parts. And they're powered by an 18650 single cell. And it's transmitting to a Google API to give me this feed every 20 minutes. And this is really just the story of this composting process. It took 72 days. Initially, because that carbon nitrogen ratio was nearly ideal anyway, I'm just experimenting as I go. I added a tiny piece of urea. It's so high in moisture that more than likely volatilized really quickly and my temperature's fallen off again. I need to hit this thermophilic range. It's 45 degrees. That's where your sterilization and whatnot happens, and the nitrification of ammonia into nitrates. And this next step here, I've added a small piece of urea again and a bulking agent, which was just 2 kilos of sawdust. And I've produced a smaller blip. And I'm getting pretty nervous now because it's getting close to May and I still haven't really done anything to produce a result. So I filled up the remaining volume of these tumblers with as much sawdust as I could and added a source of organic nitrogen that's not going to volatilize, which gave me this large spike. I still haven't reached a thermophilic range. And the whole time here, we're turning every 24 hours to homogenize the mix, and that was what literature suggested would give the fastest decomposition. And this is tapered off. And finally, towards the end of June, I just had to pull the material out anyway, because I needed to try and grow something in it. And this is the result here. So to finalize composting, you want to check the stability and maturity of the compost. And you do that with the germination assay, and that's just based on the compost extracts germinating a seed, and you're measuring the root length, which will give you these red bars here, which is your germination index. I didn't come anywhere near getting something that wasn't phytotoxic, so I knew the growing was going to be quite nasty. And I've chosen at this point to remove one of the treatments. The initial plan was to basically T2 and T3 have a 5% weight on weight biochar added to their system to basically try and physically absorb some of these heavy metals that are leaching. And T2 and T3 are exactly the same, and the germination index is very different between the two. So I scrubbed out T3. And, basically all the experimentation that occurs from here on out is only with T2. Yeah, so this is a nitrogen and phosphorus budget for the growing trial and remaining nutrients in the substrate. So our red bars can't really be directly compared to the green and blue, as it's a different species growing in the initial treatment has no biochar added to it. That species is a brassica. It's a hybrid accumulator of heavy metals. The green bar is an inoculated rye. It's inoculated with the Rhizophagus irregularis, which has a mycorrhizal or symbiotic association with rye. And the blue is rye again, but it hasn't been inoculated. And these little numbers above there are the results of each of the growing trials. So I'm just focusing on the green and the blue. 1.9 grams on a dry matter basis of rye was produced versus 2.6 from the non-inoculated rye. I wanted to see from that system as well, we can see what we saw then, what remained in the substrate that was left over after growing. And I ran 2 or 3 leachate analysis at the sowing of the growing trial, and also another one at the harvest, just to see how much that substrate is going to be letting go of nitrogen and phosphorus. And you can see probably the big comparison here is the addition of biochar and that surface adsorption of fixing a lot of that nitrogen and phosphorus in place, whereas that T1 treatment that has no nitrogen or phosphorus in it has just let it all go, basically. I also wanted to test for hexavalent chromium leaching and leaving the system. I don't know if you can see these plants here, but they all look sick. They're all growing in poison, basically. But the T1 treatment again lost 51 micrograms of hexavalent chromium in that first leachate analysis, as compared to 7 and 8 for an uninoculated rye. And by the time I did the second leachate analysis at the end of the growing trial, which was 6 weeks later, there's nothing left. And the EPA TAS limits, which I still am confused about, and they don't list, it's basically not applicable for chromium or hexavalent chromium leachate. So it defaults to a max total concentration in the actual soil, which is 1,000 micrograms per kilo. And I found 2 other standards. The Italian standard was 5 micrograms per litre, so it's a different unit, but this still seems high to me. And the regulator in China, theirs was 50 micrograms. This is probably like, this is very satisfying for me to look at. I don't know why. It's the partitioning of the chromium between the foliage roots and the substrate. So I'm trying to follow where it goes potentially. There's literature that has different management methods where they come through and they'll mow or whatnot to remove those metals out of the system. But we've got our T1 hyperaccumulator there. Our inoculated rye is the green and the uninoculated is the blue. And starting on the left, this is the chromium in the foliage. So you suggest that the mycorrhizal fungi has translocated that into above ground parts, which is very cool. And it's also held more in the roots as opposed to the hyperaccumulated and the untreated rye. And the treated and untreated rye have a similar amount remaining in the substrate. So the mycorrhizal fungi looks like it's made better use of that chromium, but possibly at a cost of better plant development. So in conclusion, the 150 litre rotary composting method's not fit for purpose, more than likely due to a lack of thermal mass. The Rhizophagus irregularis inoculated SPSM appear to partition more chromium than the non-inoculated, which might provide some interesting options for nurseries in the future that may be inoculating radiata, and they could be look at potentially using some ectomycorrhizal fungi to assist their growing if they want to use this substrate. The biochar appeared to have reduced the leachate, which was good. And the basically further investigation is required to assess the suitability of SPSM in production nurseries, and that I really need replication in the baseline treatments to help describe any variability, because obviously results are quite variable, but the material and resources required for that need a larger scale. So larger volumes, hotter longer, and give you a more stable and mature compost at the ends. The growing trial actually needs to use radiata. And yeah, as I mentioned before, using like a nectar-mycorrhizal fungi as an alternative inoculant may provide better, or it might be a worse biomass result than what we saw earlier. That's it. I just want to thank Rich and Dougal for helping me out this year and being such a good source of support. And references per page are on the back, and this is the little board that I made to do the monitoring. I sort of and again, yeah, Timberlands, thanks so much for helping me with the budget and also some expertise.
Any questions? Nathan?
Yeah, Chaz, really nice project, really interesting and important stuff. I've a very simple question. How did you measure the chromium? What method did you use for measuring the chromium in the plants or?
In the actual plant tissue itself. What's the word? ICP-AES, like instrument coupled. I can never remember how that's pronounced, but Analytical Services, TAS, helped me out with that one. So,
Cool. Any other question for Chair? Yes, Dougal.
Chaz, if you were designing that again, would you consider greater use of biochar?
The literature sort of suggested you get a diminishing return after that 5% mark. There was some evidence that suggested 10% weight on weight was also good, but I was trying to get the most bang for the least amount. So thinking it at scale down the road.
I was just wondering when you'd spoken to Timberland specific about doing a scaled up trial and whether they're interested in it based on the results that you've got, because obviously you're developing a technique.
Yeah.
Are they willing to take it to the next step and trial it? The scales you're pointing to?
Yeah, I think everyone in forestry, I think at the moment is pretty keen to do as much as they can to develop more sustainable methods. And their partner, Hills Nursery, up on the northwest-ish, they're definitely keen to see things happen at scales. So,
One more question, sorry. So was it something about the rotary compost which wouldn't get up to the thermal mass, or is it something about the forestry product which is not able to get the bacteria up to that level?
Yeah, it's just it's how saturated it is, so it's very anaerobic in there. It's to start that nitrification, you need to add a lot more bulking agent to it. But I'm wondering if it was a 500 litre tumbler, whether or not you'd need that, or you could get that going just by tumbling. Maybe some sort of servo motor to just continually slowly move it might oxygenate it enough to not have to add anything. Cheers, thanks.
Thank you very much. I think that's the end of all the presentations. So thank you everyone for coming. I really appreciate your coming out to support all our students, and I truly believe that they're also appreciative of that.